couple things about this because it does some funny things under unusual situations, nothing you'll typically run into. But this isn't big. Of the 760 millimeters of total pressure you have when you're in this room, only 47 is water vapor. That's not a huge percentage, right? It's pretty small. What is it? Not even 10%. So it's not a huge deal at sea level. But here's what's cool with water vapor. Every other gas we look at, if you change the pressure, you know, what's going to happen to the partial pressure is it changes also, right? If total pressure goes down, the partial pressure of oxygen goes down as well, right? Water vapor works, I'm going to say, a little differently. The partial pressure of water vapor, assuming the air is 100% saturated with water vapor, so what you find in your lungs, this partial pressure is not determined by atmospheric pressure. It's determined by temperature. And your body temperature is always 37 degrees Celsius, right? So the reason I say this is interesting, right now at sea level, the partial pressure of water vapor in your lungs is 47 millimeters of mercury. If you went to the top of Mount Everest, guess what the partial pressure of water vapor is in your lungs? 47 millimeters of mercury. That's almost constant. As long as your body temperature is the same and the air in your lungs is saturated, which it will be, partial pressure of water vapor is always 47. And the reason that's interesting is at sea level, 47 millimeters of mercury is not that big a fraction of the total pressure, is it? 47 millimeters versus 760. But what if you go up in elevation high enough? In fact, what if you went up in one of those, the newer supersonic transports they're making, you know, they go up at 50,000 feet. If you went up in an airplane to 50,000 feet and it depressurized, if the total atmospheric pressure is 47 millimeters of mercury, guess what the partial pressure of water vapor in your lungs is going to be? 47 millimeters of mercury. So if you were, for some reason, up at 50,000 feet, what would be in your lungs? You only got 47 millimeters of mercury, and that's the partial pressure of water vapor, which is what you'll have in your lungs. You're just going to be full of water vapor. You look like you're ventilating. You'll go, but you know what's really happening? You're just outgassing water vapor. You're not getting any air in your lungs. Of course, you die at 50,000 feet because there's no oxygen there. You die at 29, you're certainly dead at 50. But what I'm trying to get at here, as you go up in elevation, that proportion of sort of pressure in your lungs taken up by water vapor stays the same, and it becomes a bigger and bigger and bigger fraction of what's in your lungs. At the top of Mount Everest, it's getting close to 25% of the air in your lungs is water vapor because the total pressure is so low. And it's related to, you cook some. If you look in your cookbooks, do you notice there's separate recipes for cooking or canning as you go to high altitude? Right. If you're above 2,000 feet, you'll cook for an extra so many minutes, right? Is that ringing a bell at all? Your recipes? You do recipes, man. Just can. Because the boiling point of water changes, it's not fixed. At sea level, the boiling point of water is 100 degrees Celsius, right? But as you reduce the pressure, water boils at lower and lower temperatures. It's somewhere around the lower 50,000 feet, the boiling point of water is 37 degrees Celsius. So literally what's happened to you when you get high enough up in the atmosphere, the water's just boiling off your lungs. You're just filling your whole respiratory system with steam. There's no way to get any other air in there. Anyway, so there are two things that are affecting this drop in oxygen just by inhaling the air. Most of it is just the inefficiency. You're mixing fresh air with a bunch of old stale air that's already lost its oxygen, but you're also diluting the oxygen because you're adding a lot of water vapor to it that wasn't in the atmospheric air to start with. All right, so this is the partial pressure of oxygen in the alveoli. Now that is going to move by diffusion into your blood. Now admittedly, we're looking at pulmonary veins and systemic arteries, but as far as the PO2 is concerned, there really isn't going to be any difference because the pulmonary veins bring it in the heart, nothing happens to it, it's pumped back out. So this is typically arterial pressure. So if you look at the capillaries as they're leaving the lungs, so what's kind of collecting the pulmonary veins, which are going to be systemic arteries. There's not much of a drop there. In fact, as I mentioned, oxygen diffuses from the alveoli of your blood, and you basically reach equilibrium. It's very efficient as far as diffusion is concerned. Yeah, you might have noticed a slight drop from 104 to 95, that's not due to inefficiencies in diffusion. You remember when I talked about ventilation perfusion matching last time? You'll always have portions of the lung, that's never 100%. You'll always have portions of the lung that aren't ventilated, that a little bit of blood still goes to. Well, of course, that blood reaches equilibrium, but it reaches equilibrium with some of that old stale air, so it's going to have a lower PO2. By the time you mix all that blood coming from the lungs, you might see a slight drop in PO2. For all practical purposes, they're the same. The PO2 in your alveoli is pretty close to what the PO2 is going to be in your arterial blood. And in fact, if you're going in for kind of more thorough blood workup, they will measure the PO2 of your blood. They have to draw the blood specially. You can't take it out of veins. They do an arterial draw. It has to be gas tight. But they'll plug it in a machine and they'll read your PO2. PO2 in arterial blood is high. It's going to be somewhere close to 100 millimeters of mercury. 98, 95, 100. That is the blood that is going to be moved from your lungs down to your tissues. And of course, the reason we're moving oxygen to the tissues is you need it for cell respiration. All of this oxygen, it's going to diffuse out of the blood into the tissue cells, into the mitochondria, to act as that electron acceptor for cell respiration. So the tissues are actually where you're using the oxygen. So if you take a sample of fluid around metabolically active tissues, you're going to have a PO2 that averages about 40 millimeters of mercury. Which you would expect. You're going to get the lowest PO2 with the tissues because that's what's consuming or using the oxygen. And a couple things I want to mention. This is an average. There's actually a wide range. In really heavily metabolically active tissue, the PO2 in real active tissues could drop to as low as almost 15. In tissues that are not doing much of anything, the PO2 might be about 7. But if you pull it, on average, the blood, the PO2 around all of your tissues combined is about 40 millimeters of mercury. And as the blood passes through those tissues, through the systemic capillaries, of course, oxygen can diffuse and reach equilibrium. And by the time you collect the blood at the venous end of these systemic capillaries, you'll see the PO2 is the same as it was in the tissues. Venous blood will have a PO2 of about 40 millimeters of mercury, and that is what will circulate back up into the pulmonary capillaries. So now I want you to note one thing here. What is the concentration gradient or partial pressure gradient moving oxygen from the alveoli into the venous end of the pulmonary capillaries? So you have 104 is a PO2 in the alveoli. The venous blood coming into the pulmonary capillaries is 40. So we call it the delta PO2 equals, what is that, 64 millimeters of mercury. The difference in partial pressure basically between the venous blood and the air in the alveoli is 64 millimeters of mercury, right? That's the partial pressure gradient or concentration gradient pushing the oxygen, if you will, into the blood. Now remember that, because now we do the other gas and it's a little different. The other respiratory gas is carbon dioxide. Talk about it too much yet. It's moving the opposite direction, but you can measure the concentration of carbon dioxide as well using partial pressure. So let's go back to the top. And I'll do the same thing just with CO2. I'll go through it kind of quick because it's not as critical. First of all, in the atmosphere, there's very little carbon dioxide in atmospheric air. So we're talking about 0.3 millimeters of mercury. Approximately none as far as respiratory system is concerned. There's not much CO2 in atmospheric air. Now we take that air, we inhale it, look at the alveoli. Huge increase. 40. There's actually, compared to what's in the atmosphere, there's a huge amount of CO2 in your lungs. Now there's a reason for this, we'll get into it eventually. You actually control your breathing to retain carbon dioxide. As we'll see in a bit, you don't even breathe to control oxygen levels. There's so much oxygen in the atmospheric air, it doesn't matter. You're breathing almost exclusively to control CO2 levels and maintain real high CO2 levels in your lungs. So So that's why it gets so high. You're breathing to keep that number high. But again, just like with oxygen, as the blood moves through the pulmonary capillaries, it'll reach equilibrium with the CO2. So the PCO2 in the arterial blood leaving the lungs is the same as it is in the alveoli, about 40 millimeters of mercury. That's what you have in the arteries. Then moving down to the tissues. And of course, the tissues are the source of the carbon dioxide. They're produced in the metabolic pathways. So PCO2 is higher in tissues. It's about 46 millimeters of mercury. And then finally, that will reach equilibrium with the venous blood. It'll be at 46 millimeters of mercury. And that venous blood will go back to the lungs. Bubble, 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 end up back here. And the CO2 will diffuse out. So it's moving in the opposite direction of the oxygen. It's being produced in the tissues and moved to the lungs. The oxygen is just reversing. But here's what I want to look at next. All right, if you compare the PCO2 in the alveoli, to the PCO2 in the venous blood, down here at the bottom, what is the difference in CO2 sort of pushing the CO2 out of your blood into the lungs so you can exhale it? PCO2 of 40 here, and this is the venous blood, it's a PO2 of 46. The partial pressure gradient is 6 millimeters of mercury, right? That's the difference? Nod your head. Right, that's what it is. I mean, you can pull gas samples out in these various places, and that's what you would measure. But now here's my question. Concentration gradient, does that affect rates of diffusion?
Yes.
So here's my question for you. If I have a partial pressure gradient of 64 millimeters of mercury, sort of pushing oxygen from the alveoli into your blood, and I have a partial pressure gradient of 6 millimeters of mercury, pushing CO2 from the alveoli, or from the blood into your alveoli, which do you think you move more of, in a minute of time? Do you think more oxygen diffuses across the respiratory membrane or more CO2? More, if I look at those numbers and have to guess, yes, more oxygen seems to make sense intuitively. However, it's one of those questions where, no, that's not right. But it does seem weird. That's not talking about it. You've got, what, 10 times the concentration gradient of oxygen as opposed to carbon dioxide, right? So just look at those numbers. Yeah, I should move more oxygen than CO2. You don't. You move, for all practical purposes, you move about the same amount each. And the reason for that is you are using all of this oxygen in cell respiration. You are producing all of this CO2 in cell respiration. It's not that you necessarily need to write this down, but this is that net equation for cell respiration when you're doing glycolysis and the Krebs cycle electron transfer chain. It should look just like what you're looking at. What I want you to note is when you burn a glucose molecule in these metabolic pathways, you use 6 oxygen molecules. When you burn a glucose molecule in these metabolic pathways, you produce 6 CO2 molecules. It is 1-to-1. For every oxygen molecule that goes into your cells, it's going to produce 1 CO2 molecule. Because that's the only place they're going to and coming from. Does that make sense? Which means if a liter of oxygen moves from my alveoli into my blood, then about how much CO2 needs to move from my blood into my alveoli? About a liter. They have to be about the same. Now this is making a couple assumptions. One, the respiratory quotient is 1. I'll talk about that in a minute. But basically, if you're just consuming glucose, yeah, this is a 1-to-1 relationship. So you have to move the same amount of oxygen across the respiratory membrane as you move CO2, just in opposite directions. Does that make sense? How is that possible when you have this huge difference in concentration gradient? I will tell you, it is true, you move about the same amount of CO2 and oxygen. But again, you've got such a huge concentration gradient. How is that possible? That leads me to the last thing we'll look at today, which is solubility.
I have a question.
Yes?
How did you get the changing pressure to 64 millimeters?
Oh, 64?
Yeah.
You have venous blood has a PO2 of 40, right, coming into the lungs. In the alveoli, the PO2 is 104. So what you basically have, here's the alveolus, PO2 is 104. Here's the pulmonary blood vessels here, PO2 is 40. The difference in concentration allowing the oxygen to fuse in is 64, right?
You subtracted, how did you calculate it? How did you calculate it to get like 64?
To subtract the difference.
You do the same thing for the...
Yeah, because here for O2 or CO2, you've got 40 millimeters of mercury in the alveolus. You have 46 in the blood, so there's 6 millimeters extra pressure in the blood, allowing it to diffuse into the alveoli, right?
Yeah.
That's all. But it's 10 times higher for oxygen. Now, the reason you get away with that, you know about solubility, right? Solubility is one of those things, we all know what it is, but it's actually kind of really hard to explain. You know, it might be easier with solids and liquids since we're more used to them. I mean, if I take a teaspoon of sugar in an 8-ounce cup of water, and I put that teaspoon of sugar in there and stir it up, what will happen to sugar? It'll all dissolve. I take a second teaspoon, dump it in that 8 ounces of water, stir it up. Will it dissolve? Probably. How about a third? Fourth? Fifth? Sixth? Can I keep putting sugar in it? Will it keep dissolving? Forever.
No.
I can put 5 pounds of sugar in an 8-ounce cup. No, what eventually happens? You saturate the solution, and then you can't get any more sugar to dissolve, because if more dissolves in, some precipitates out, and you get crystals forming in it, and that's it. It's a saturated solution, right? But we agree sugar, at least in a relative sense, is quite soluble. That is, it takes a lot of sugar to dump into water before you're done, before you can't get any more in it. Agree? How about calcium carbonate? That's like the limestone rock out here, or the agricultural lime you throw in the farm fields. Is that soluble in water? If I took some pulverized limestone, put it in 8 ounces of water, stirred it around, would any of it dissolve? The answer is yes. They don't have water heating problems around here where they like fill up with this white stony junk and your pipes get all. You've been into the Mammoth Cave, frozen Niagara. All those cave formations, how do they form? Rainwater traveling from the surface here, it dissolves limestone. And then when it comes into the cave, the water evaporates and it deposits that limestone. Or if you ever, not that many of you do this, but like taking a pot of water to boil on the stove and forget you had it on there and it all boils completely out.
Oh my God.
And then what does the inside of the pot look like? It's got this white, hazy junk on it. That's calcium carbonate. See, calcium carbonate is soluble in water, but if I took 1 teaspoon of powdered limestone, dumped it in 8 ounces of water, it would not dissolve. It's got less solubility. So really, solubility is how much of a substance you can dissolve in the fluid, right? Does that make sense? We kind of know what that is. Gases have different solubilities. Just like calcium carbonate and sugar, gases have different solubilities. So let me show you how I'll demonstrate that with this stuff. All right, what I'm going to do, I'm going to take a container, kind of like a little aquarium, and I'm going to half fill it with water, and I'm going to connect it to an oxygen tank. So all I'm going to do, I'm going to introduce oxygen until the partial pressure of oxygen in this gas is 100 millimeters of mercury. Now initially, there's no oxygen in the water. So the PO2 is 0 in the water. So the oxygen will diffuse from the gas phase into the water. And when will the oxygen stop diffusing into the water? We have equal partial pressures, right? That's what equilibrium is. So the PO2 equals 100 millimeters of mercury. That's equal partial pressures. Diffusion will stop. But answer me this. Where is there more oxygen? Is it in the top half of the box, which is all oxygen gas, or is it in the bottom half where there's water as well? The partial pressures are the same. Does that mean there's an equal amount of oxygen in both halves? The water versus the gas phase?
No.
There is not. Because, again, it's solubility. Every time oxygen dissolves into the water, it exerts a partial pressure. As we'll see, oxygen does not have much solubility. You really don't get very many oxygen molecules dissolved in the water, and the PO2 is 100. And again, what that means is if another oxygen molecule dissolves in the water, one will pop out, so you've reached equilibrium. Now imagine I do exactly the same thing, except this time I'm going to have the same setup, same half filled with water, but now I'm going to connect this to a bottle of CO2. And I do the same thing. I introduce CO2 till the PCO2 equals 100 millimeters of mercury. Again, the CO2 will dissolve into the water until the PCO2 in the water phase is the same as the gas phase. Now, here's the only other bit of information you get, because we're not looking at real absolute numbers here, it's sort of qualitative. CO2, 20 times more soluble than O2. Carbon dioxide is very soluble in water compared to oxygen, about 20 times more soluble. So what does that mean? All right, these have all reached equilibrium. Now let's just count these dots for a minute. You got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Let's say there's 10. Those dots represent the number of oxygen molecules that diffuse into the water when you reach equilibrium. If we do the same thing with CO2, because CO2 has 20 times the solubility, I'm going to add
Did he just count 100? Oh my god.
That's the difference. You'll still get oxygen moving into the water phase here, but it's got such a low solubility, not much gets in there, and the PO2 pops up to 100. CO2, because it's 20 times more soluble, 20 times more CO2 molecules dissolve in the water before you reach that PCO2 of 100. So even though partial pressures are all the same across the board here, I have 20 times more CO2 molecules dissolved in that half of the tank of the water. Does that kind of make sense? That gets us back to the blood here. Blood, we're talking water. The plasma is basically water. It behaves the same way. The reason PCO2 has such a high partial pressure gradient is because oxygen has such a low solubility. In order to get the same amount of oxygen into the blood, you need a huge partial pressure gradient. Carbon dioxide is so soluble, a small partial pressure gradient allows a ton of it, many molecules, to move across the respiratory membrane. This is called Henry's law. We're not going to stick any numbers on this one like Dalton's. But if you want to know how much of a gas you can get into a solution, into water or plasma, there's two components. One is the partial pressure. All else being equal, the higher the partial pressure, the more of that gas will go into solution. But second is the solubility. So the two things that determine how much of a gas will go into solution are partial pressure gradient and solubility. And solubility is a complicated variable. It's affected by everything. The type of gas, the type of solution, any ions in the solution, temperatures, pHs, almost anything that affects solubility. It's pretty complicated. But two factors that affect how much of a gas will go into solution: partial pressure and solubility. And again, so the reason you don't need much of a partial pressure gradient for CO2 is because of the high solubility. You're actually moving about the same amount of oxygen in CO2. It's just with the high solubility, a millimeter of mercury difference in partial pressure causes a whole lot more CO2 molecules to move across the membrane as opposed to oxygen. Having a fun, exciting, but not too exciting Thanksgiving. I have put digestive anatomy videos up, respiratory system videos up. I also have a narrated PowerPoint on the last of the slides. I meant to get to it last time, but I had to leave early. So you can look at those as well. It's just a scattering of slides. Some on blood vessels, some on the liver, some on intestines. Just two. It's got random slides on it. They should all be up on Blackboard. Exam is
Monday.
the next time I see you. So look forward to during Thanksgiving.
Which is over just the heart.
Lab exam is not the first. We're moving it to the 6th. Is that all right with you? You're going to a little bit more time. See, a little cramp. So March and I got together. I think we can push it off. Basically, what, the next Monday or something?
Yeah, that's a lot.
Okay, so this coming up Monday.
Is the lecture exam.
Right.
Which covers-
What's it over?
29th is lecture exam. It's circulatory system.
So the 29th.
Everything. So the
The
electrical,
6th.
everything
The
over the heart,
6th is lab
cardiovascular.
exam, which covers white blood cells, respiratory, digestive, and renal. We haven't done renal yet. We're doing that Monday.
December 6th.
December 6th is what we're trying to do for it.
Digestive, renal, and respiratory, right?
That is blood cell identification, respiratory, digestive, and renal. Get your time in to study that junk because it should be a fairly easy exam compared to the first one.
But the exam we are taking on the Monday, it will be only
Circulatory. blood vessels, heart, blood pressure, anything. It won't have respiratory system, but anything on circulatory is fair game.
What about the female reproductive?
That was on the last exam, I think.
What, like 50 questions?
Something like the last one, yeah. 50 or
No.
so, multiple choice, fill in the blank, 4 to 6 short answers. Short answers, just to give you an idea. Some quick ones. Compare cardiac muscle cells to skeletal muscle cells. That's been a common question. Compare arteries to veins. Yeah, that's kind of a good question. Describe or label an EKG, a basic lead to EKG. That will be quite likely on there.
So just the P waves, the QRS, the
T
T.
wave, what they're measuring electrically and what they are related to mechanically.
The main difference between cardiac and skeletal is just the branches. Cardiac has branches.
Oh, there's way more.
Yeah.
Cardiac has sodium
less
leak
SR.
channels.
Yeah.
Cardiac muscle cell has gap junctions. Cardiac muscle cell has less sarcoplasmic reticulum. Cardiac muscle cell is myogenic, the leak channels. Cardiac muscle cells are only aerobic. Skeletal muscle cells are both. Bigger T tubules in cardiac muscle cells, they're branching. They have a intercalated disc. There's a bunch. Some of them are related, but there's a bunch of things.
What was
He said cardiac muscle.
The what, huh?
Label EKG.
Lab exam is blood cells, red blood cells, platelets, and then a neutrophils, eosinophils, basophils. That's it. That's one of the labs. So I'll have scopes out and identify this blood cell. What's that?
like all was on
Blood cell identification, respiratory system,
What else?
digestive system, and renal system. We have not done a renal system yet. There's a video up with the respiratory system models, the respiratory system on a cat, and I believe the cat's digestive system. There's some videos posted if you want to play with those over the weekend.
Thank you.
Just so you're not bored.